ongoing potentiation in the stratum oriens for control slices that receive stimulation to the Schaefer collateral inputs to the stratum oriens, either with no other treatment, shown in blue, and in sections that received emetine in the stratum radiatum. In other words, local application of the protein synthesis inhibitor emetine into the stratum radiatum produced a decay in LTP for stimulation of the stratum radiatum, but did not affect potentiation of the stratum oriens. The two graphs at the right show, I guess, kind of the inverse effect. If emetine is applied to the stratum oriens, it selectively disrupts potentiation of inputs to the stratum oriens without disrupting LTP of the stratum radiatum. Now, keep in mind that emetine is one of the antibiotics that I mentioned a few slides back, which blocks translation of mRNA to protein rather than transcription of DNA to RNA. So the results of this set of experiments on plasticity of the stratum radiatum and stratum oriens suggest that local application of a translational inhibitor of protein synthesis can block potentiation of synapses locally, restricted to the area where the synthesis inhibitor was applied. In addition, Bradshaw and colleagues showed that administration of emetine into the soma failed to block LTP in the stimulated dendritic fields. So taken all together, these data suggest that late LTP, the first phase of it, requires local translation of mRNA that does not involve signaling to the soma or nucleus. But those experiments don't conclusively tell us where the mRNA actually comes from. Of course, the time course suggests that late LTP starts too early for it to involve genomic signaling, as signaling to the genome would take at least 2 to 3 hours, and then the cell would need to transport the newly transcribed mRNA out to the dendritic field. Moreover, we already saw an experiment that showed that the translational inhibitor, anisomycin, had a detrimental impact on LTP at an earlier time than did the transcriptional inhibitor, actinomycin. If transcription of RNA was required to support the local protein synthesis, then there actually should not have been a difference between the timing of the effects of anisomycin and actinomycin. Nevertheless, Catherine Dickinson in David Wiley's lab did an ingenious experiment in which they autotomized the dendritic fields, meaning that they isolated them by cutting them away from the soma. If they isolated the apical dendritic field in the stratum radiatum before stimulating the Schaefer collateral inputs to the stratum radiatum, they still got potentiation that endured for more than 3 hours in the stimulated synapses. This demonstrates quite conclusively that the local synthesis of proteins from pre-existing mRNA supports late LTP in hippocampal neurons. Okay, so clearly the first part of late LTP depends only upon local synthesis of proteins from messenger RNA that already exists in the dendrites nearby. But I showed you evidence from the experiment comparing the effects of anisomycin and actinomycin that later stage late LTP requires genomic signaling, because actinomycin, the antibiotic that blocks transcription of DNA to RNA, blocks that later stage. So our next questions that you need to ask are, well, what signals the nucleus to start transcribing genes in support of late LTP? And how do these signals get to the nucleus? So the answer is that the signals must involve second messengers and secondary effectors, like the ones we saw in Chapter 3, along with transcription factors like CREB. So that's at least a preliminary answer to the question, what signals the nucleus to start transcribing genes in support of late LTP? It's the cascades of signaling, and we'll see more about those cascades in a minute. Then there's the question, how do these signals get there? For that, we actually have two models. In one model, which is shown in the upper diagram on this slide, potentiating activity at an individual synapse activates cascades of signaling, as we've already seen. An example of signaling in this model is that adenylate cyclase generates cyclic AMP, which frees the catalytic subunits of protein kinase A from the regulatory dimer. Then, in addition to local phosphorylating actions at the activated spine, some of the protein kinase A is transported to the nucleus, where it activates transcription factors like CREB, the cyclic AMP response binding protein. This is known as the synapse to nucleus signaling model. And in the illustration, the signals, like protein kinase A, are depicted as little green blobs heading down the dendrites to the soma and even into the nucleus. The other possibility is that the depolarization at activated spines causes depolarization at the soma, and this depolarization causes voltage-gated calcium channels to open, providing an influx of calcium that activates secondary effectors like CAM kinase 4 and MAP kinase, which also phosphorylate transcription factors like CREB. This is known as the soma to nucleus model, and it's shown in the lower diagram on this slide. In this case, the calcium influx is shown entering the soma, where it activates calcium-sensitive proteins like calmodulin, and even perhaps into the nucleus to activate proteins there. In both models, the genomic signaling must produce outcomes that will impact the activated spines. These are known as plasticity products, that is, mRNA or protein, and they're transported back to the activated spines. In our two diagrams on this slide, the plasticity products are shown as little purple blobs. We'll see more details about the specific plasticity products in the next chapter. For now, let's focus on the signals heading to the nucleus. On this slide, I've placed a diagram that I modified from your textbook to isolate two of the signaling pathways for the synapse to nucleus model. The diagram in your textbook on page 91 shows both synapse to nucleus and soma to nucleus mechanisms on the same diagram. I just deleted the soma to nucleus pathway so you can clearly and specifically see the synapse to nucleus model. Now please note that the diagram actually represents receptors out in dendritic spines and signals that would convey all the way to the nucleus along the dendrites, even though the diagram looks like the receptors are like sitting nearby on the soma. If I was making your textbook, I would have drawn that much more clearly. But I'm certain that you can visualize the G protein coupled receptor and the tyrosine kinase receptor sitting on a spine, and the arrows in this pathway extending all the way down dendrites into the nucleus. Okay, on the previous slide, I mentioned that one of the potential pathways for the synapse to nucleus model originates with activation of adenylate cyclase. Of course, we know that adenylate cyclase can be activated either by calcium influx or by signaling through G proteins associated with a metabotropic receptor. The cascade, starting with a G protein coupled receptor through cyclic AMP to protein kinase A, which is then translocated to the nucleus, is shown in the upper left part of the large diagram on this slide. Now, inside the nucleus, the protein kinase A phosphorylates CREB at serine 133, and CREB binds to the DNA and initiates a cascade of genomic signaling to eventually induce transcription of a variety of target genes, including genes that encode enzymes, structural components, receptors or ion channels, and so on. The latter stages of this process are depicted in the lower part of the large diagram, and the plasticity products that are generated could be messenger RNA or proteins that can, either one of them, that can be transported back to the activated spines. I'd also like you to note the similarity between this pathway and long-term plasticity in the Aplysia gill siphon withdrawal response. Remember how I described that protein kinase A was implicated in turning on transcription of genes that are implicated in restructuring of the axonal arbor in Aplysia? Well, in Aplysia, that function actually occurs when their protein kinase A activates a protein called Aplysia CREB, or ACREB. So, across eons of evolutionary time, a very similar cascade of signaling is retained in the invertebrate Aplysia and in mammals, including humans. Of course, if the mechanism is conserved across all these evolutionary adaptations and all the species that have derived from some primordial ancestor we have in common, you must imagine that this is a very important and essential mechanism. I also need you to note that the pathway from adenylate cyclase to CREB is not the only one that can produce activity-dependent synapse to nucleus signaling. There are many additional cascades, including signaling through tyrosine kinase receptors. Now, some people call them TRK receptors, and some people call them TRAC receptors, but they are, of course, the receptors that are activated by growth factors, and they signal by phosphorylating tyrosines through kinase activity in the intracellular domain of the receptor. These receptors are activated by growth factors, and yes, growth factors play an important role in synaptic plasticity. We'll be looking at the role of growth factors in plasticity a little bit in Chapter 6. Now, in this illustration, brain-derived neurotrophic factor, or BDNF, a growth factor that is really important in the support and differentiation of hippocampal neurons, activates the TRKB receptor. That induces phosphorylation of the second messenger RAS to then activate ERK kinase, not shown on this pathway, that phosphorylates mitogen-associated protein kinase, or MAP kinase. The activated MAP kinase then translocates to the nucleus to phosphorylate CREB at serine 133, the same serine that's phosphorylated by protein kinase A, and then the cascade of gene transcription and generation of plasticity products ensues. And this diagram represents the soma to nucleus model. Once again, I modified the diagram from the textbook to isolate the soma to nucleus pathways, and I removed the two synapse to nucleus pathways that were on the previous slide. Now this time you don't have to visualize any distortion of the diagram, because this time the mechanism, the voltage-gated calcium channel, is actually right here in the soma. Now in this model you can see that calcium influx through voltage-gated channels activates at least two signaling pathways that converge on phosphorylation of Krebs. One pathway is where somatic influx of calcium activates calmodulin, which then reaches and enters the nucleus. And then once inside the nucleus, it activates CAM kinase 4, which phosphorylates serine 133 on Krebs to activate genomic signaling. Now your textbook also describes that calcium can get all the way into the nucleus and activate nuclear calmodulin that will in turn activate the nuclear CAM kinase 4. There certainly is nuclear calmodulin, so I won't argue the point with your textbook author, but I don't know the specific evidence for this slightly different version of the calcium to calmodulin to CAM kinase 4 pathway. So I'm not going to worry about that pathway, but I just wanted to bring it up because your textbook does describe it. Now then, the other pathway is where the influx of calcium activates MAP kinase, which then enters the nucleus to phosphorylate serine 133 on Krebs and turn on Krebs function. We also need to note that these pathways from adenylate cyclase to protein kinase A to Krebs, or from TRK receptors to MAP kinase to Krebs, and from voltage-gated calcium channel opening through calcium calmodulin and CAM kinase 4 or through MAP kinase to Krebs, are all essential components of late LTP. Because if you block either protein kinase A or MAP kinase, specifically at the soma, you will block late LTP, and you'll also decrease Krebs-dependent protein expression. And there's lots of additional evidence to support these models, but I'm going to focus on the soma to nucleus model for a few minutes. It seems that these soma to nucleus signaling pathways are particularly important because there's actually a need to initiate genomic signaling very rapidly. The cascade of signaling from kinase activation to phosphorylation of Krebs to increases in expression of downstream gene products that are implicated in synaptic potentiation can take a few hours. And the experimental evidence I've previously shown you clearly establishes that early LTP decays within a few hours. So we need to activate mechanisms that will support the potentiation starting as quickly as possible. If the signaling to the nucleus was entirely dependent on synapse to nucleus signaling cascades, that would introduce an even longer delay as those signals are slowly transported to the nucleus down the dendritic arbor. Therefore, some scientists have argued that those signals might not really be important in the initial stages of the genomic signaling, and that rather the soma to nucleus model is more important in activating that genomic signaling. Furthermore, antidromic stimulation of the axons of CA1 pyramidal cells timed together with weak Schaefer collateral stimulation actually produces long-lasting late LTP. So consider the graph on this slide. Bucalo and collaborators provided weak tetanic stimulation of Schaefer collaterals. In some preparations, they paired the weak tetanic stimulation with antidromic stimulation of the CA1 pyramidal cell axons. In other words, they provided enough stimulation of those axons so they fired action potentials. In the other preparations, they simply administered the weak stimulation of Schaefer collaterals. And of course, after these stimuli were administered, they resumed test pulses. The lower plot on the graph shows the responses of the preparations that only received the weak tetanic stimulation. And you can see once again that weak tetanic stimulation of Schaefer collaterals produces LTP that degrades over a period of minutes to hours. However, the preparations that received antidromic stimulation paired with the weak tetany of Schaefer collaterals exhibited robust ongoing LTP that lasted for many hours. Now, it's also known that the antidromic stimulation phosphorylates MAP kinase and Krebs. Of course, the relevance of this is that action potentials do not simply run down the axon. It wouldn't make sense that all the sodium that rushes in at the axon hillock all runs in one direction. Yes, it must go down the axon, but it also goes back into the soma, where we now all know that it will encounter and trigger voltage-gated calcium channels. This is the back-propagating action potential that I referenced in a previous chapter, and it's most certainly going to activate the soma-to-nucleus cascade of signaling to support the later phase of long-lasting or late LTP. So here we go once again. I have another opportunity to say that neurons that fire together wire together. But wait, there's still another important question regarding the fundamental function of both the synapse to nucleus and soma to nucleus models. So what is that question, you ask? Well, the question is this. If genes are being transcribed and perhaps translated at the soma to generate mRNA and/or proteins that are implicated in synaptic potentiation, how can these plasticity products be delivered specifically to the stimulated synapses and only to the stimulated synapses where they're going to be needed? Remember the principles of input specificity and spatiotemporal specificity that we learned way back in Chapter 2? Was that really only a couple of weeks ago? Less than a couple of weeks ago? Okay, so we know that LTP is restricted to only the synapses that received the high-frequency stimulation. Or in the case of weak high-frequency stimulation, the synapses that were paired somehow with strong stimulation or back-propagating action potentials. So how can plasticity products, mRNA, and proteins that are generated at the soma find just the right synapses and specifically function at those synapses to potentiate them and not at all the others? Wouldn't the plasticity products tend to go everywhere and potentiate all the synapses in the postsynaptic neuron? Does anybody have any ideas? How about this one? What if the stimulated synapses develop some kind of a synaptic marker or tag, so local transport of plasticity products would find that marker, and only the tagged synapses would be able to use those plasticity products to reinforce the synaptic modifications only at the potentiated spines. In this way, products that are generated in the soma could be transported out all over the dendritic arbor, but they're only going to be activated at those particular spines that need to be potentiated, at those particular spines that are marked to have those plasticity products targeted to them. That would work, wouldn't it? Well, indeed, it does. And it's illustrated in this diagram that I modified a bit from a paper published by Govindarajan and collaborators. I'm sure I'm butchering the name and I apologize for that. But it illustrates that stimulation of specific synapses generates potentiation tags at those synapses. These tags would then function to activate translational mechanisms to process specific forms of mRNA or to capture specific proteins as they pass by. We're going to follow up on the mechanisms of synaptic tagging further in Chapter 6. But in the meantime, I want to make certain that we're all on board with the idea that there are two overlapping waves of protein synthesis that underlie late LTP. The first is translation of pre-existing messenger RNA that is available to be used rapidly at potentiated synapses. The second is a cascade of signaling to the genome in the nucleus to activate transcriptional and translational activities on a slower timescale. The bottom line is that the basic availability of second messengers and secondary effectors at the naive synapse is not sufficient to support the continuing potentiation of the synapse after potentiating stimulation. We need local translation of pre-existing messenger RNA to support the functional and structural changes in the spine beyond the first 30 minutes or hour. Then we need genomic cascades of new synthesis of mRNA or proteins that will actually be transported out into the dendritic arbor, where they will interact with plasticity tags at the potentiated spines to supply the needed additional substrates to support the ongoing maintenance of potentiated synapses. Okay, and we're about to enter a new topic. We're about to start talking about LTP1, LTP2, and LTP3. And our time for today is nearing a close. So I think I'll pause here. And this will be continued in Chapter 5b.